Hello, hello. Oh. Hello. Hello. Okay. No questions? Everybody's good? Yes, sir. That's a good question. What chapter are we on? Is it 29? Stem cells. 29? I don't know. Yeah, I know, sorry. I don't know. Let's look. So we just switched books, so some of the stuff I didn't, switch over because I forgot. So this is at the back, right? Yes. Chapter 13. Sorry, chapter 13. Sorry. So some of the stuff is from the other textbook. So a little bit at the beginnings from the other textbook. It's somewhere in your book, but everything you need to know will be in the notes. Okay, so some of this stuff is moved around outside of how this book goes, just because I like to teach it that way better. If it's confusing, you can always send me an email and ask me where the information came from, and then I can find it for you in the new book, in our book. Okay? So we're going to start with a little bit of genetic engineering today before we get into stem cells, because it sort of leads into the whole stem cell revolution, the history of manipulating DNA, essentially, manipulating DNA for better health and well-being, okay, for us. And so what we start with is the Human Genome Project, which was what sequenced the entire genome of human beings, right? This has been now done, oh my gosh, for a long time, at least 15 years, okay? It revolutionized the ability of scientists to do better science. So when we talked about doing experiments, remember from chapter 1, making a hypothesis and doing the testing, and that if we're talking about human beings, we can't do some of the experiments that we'd like to do because it's unethical, right? It's not okay to do something we know might be harmful to people. And so we use model organisms in order to do the experiments we can't do with people. So a model organism like a mouse, right? Or fruit flies, or worms, or rats, or, nobody uses guinea pigs, but that's what everybody says, oh, you're going to be the guinea pig for the experiment, because apparently 50 years ago they used guinea pigs instead of mice for experiments, right? So that's what you'd say if somebody's doing an experiment, right? Like with you guys are all the guinea pigs for me using this new book, okay? So knowing all the DNA sequence in human beings made science move faster because we could study genes in a mouse and then find the equivalent gene in human beings. Okay? So like we said, we all come from a common ancestor, and mice are mammals, so we have a recent common ancestor. Our DNA is quite similar. We have lots and lots and lots of the same genes as mice do, right? Maybe a little bit different, obviously, because we don't look or act like mice. But a lot of our immune system, a lot of our nervous system, a lot of the physiology of people and other mammals is very, very similar, right? So the genes for insulin, right? Insulin is the protein, think of it as a hormone, that is released when you eat sugar, and it allows our cells to take in sugar out of the bloodstream to do work. Right? We use sugar, we convert it to ATP, and then we can do work. Whether that's running across campus, or thinking, or breathing, or our heart beating, we need energy to do all of that. Okay? And so insulin is the molecule that allows our cells to take up sugar. So if you don't make insulin or your gene is screwed up, you can't take up sugar from your bloodstream, and you're called diabetic. Okay, and excess sugar in your bloodstream is a big problem. Causes all kind of issues, clogs up arteries and veins and causes circulatory problems, messes up your kidneys and your liver and all your other organs. Right? So untreated diabetes is life-threatening. Okay? So because mouse insulin gene is very similar to human insulin, we can study diabetes in mice very easily, and we can just use the computer to compare what the insulin gene in mice looks like to the insulin gene in humans. Okay? So any gene that we can isolate from a model organism, we can throw into the computer. There's all kinds of crazy algorithms by those wacky computer scientists that program that stuff to find DNA that's similar to whatever you put in there. And then it gives you a similarity index to tell you, well, is it similar enough to be relevant, or is it just BS? Right? So you can do the statistics and all that crap. So you can find the human equivalent of all kinds of genes very easily now based on the Human Genome Project. Okay? Rather than having to we used to have to take the human protein, right, isolate it, and then break that down into its amino acids, and then figure out what the DNA sequence might be from there, and then try to compare it. And it took a long time, and it was a big pain in the ass. So it's much easier to study human disease based on knowing the sequence of the genomes. And now lots of different genomes have been sequenced, and genome just means all the DNA, right, in that being or in that species, so that we can just go to the database and look at comparisons of all different types of organisms to human beings and do those phylogenetic trees where we can look at who's more related to whom. Okay? And so one of the first revolutions based on the Human Genome Project and knowing sequences was something called gene therapy. Gene therapy began maybe 20, 25 years ago, and we thought it was going to be the thing that cures all diseases and all disorders. Okay? And it still has amazing potential for human health, but it ran into a whole bunch of problems. Okay? The main problem with gene therapy is that in order to transfer so the idea would be you have a defective gene, right? Let's say your gene for insulin does not work, it's messed up, it has a mutation, right? The idea would be to replace that DNA, physically replace it with good DNA from somebody that has a normal gene. Okay, we know all the sequences, we can just isolate that DNA, make more of it, and if we could get it into that person's chromosomes in their pancreas, they would then have a normal copy of that gene. They could make insulin. Okay? Ideally, that's perfect. The problem is getting that DNA into somebody's chromosome. Okay? That, as it turns out, is a little difficult. So what we do, there is an entity on this planet that is very good at putting DNA into our genome, and that is called a retrovirus. Okay? Retroviruses get in our cells. Remember we talked about last time, they're not alive. They're just a bunch of genetic material and some protein. They have to get inside your cells in order to replicate themselves. Well, as it turns out, a whole class of viruses not only get in your cells and use all your equipment, they actually stick their DNA into your chromosome. And then our cells don't know any different, and we just do transcription and translation, make proteins from it as if they're our own. Okay? So what we've done is we can take those viruses and we can remove the genes that make them bad for us, that allow them to be virulent and infect a whole bunch of cells and make you sick, and instead stick in the gene for insulin in there, replace it. And then we give that person's cells that virus, it gets in their cell, and it dumps that insulin gene into their DNA, and if all goes well, right, they make that insulin gene then. Okay. We can do this pretty well in model organisms. In mice, right, we can cure diabetes in mice doing this easily. Okay. The problem is the side effects. Okay. The problem is it's a virus, and the viral DNA stays in your cells, and sometimes our immune system says, hey, there's a virus in there, and we want to get it out. Okay? That's what your immune system's supposed to do. Right? We're not supposed to ignore viruses and say, hey, leave DNA behind. We're supposed to get rid of them. And so in some cases, the immune system targets those cells and kills them, and then that doesn't help at all. Okay. So the efficiency of this transfer and the safety is the problem generally with gene therapy. Okay. And so if we're doing it in regular cells like the pancreas, we just call it somatic cell gene therapy. A somatic cell is just a normal body cell, everyday cell. Okay. Germline gene therapy is when you actually change the DNA in an embryo. Okay, so very early. So, or in a sex cell, in the egg or the sperm. Okay, and so this is in experimental stages only in model organisms. Okay, so we can take, and we do, we can take mouse eggs and sperm, do in vitro fertilization, get that fertilized egg, and then we can replace genes in that fertilized egg doing gene therapy so that mouse now expresses some human protein as it grows in all of its cells. And then we can study what happens if this mutation in this gene is expressed in all mouse cells. Okay? That's how we found genes that cause cancer, is by, we have this, a whole bunch of people have this really nasty cancer. They're all expressing this one protein really highly. Let's see what happens if we express that protein in a mouse, and the mouse gets cancer. That gene is likely to be a cause of cancer. Okay? But we don't do this in people, right? We don't mess with DNA in people. Okay. So one success story for gene therapy is in individuals who have something called SCID, or severe combined immunodeficiency disorder. This is the bubble boy, children that are born without any functional immune system. Okay. If you don't have an immune system, you die very early within the first 6 months of life from the common cold or something silly that nobody else dies of. Okay? Right? So if your immune system is not functioning, things that just make us a little bit sick and we're annoyed by will kill you. So these individuals have to be kept in a germ-free environment or a bubble, right? A big tent. Nobody can touch them. It's filtered air. Everything that goes in there has to be sterilized, autoclaved. Nothing can have any bacteria, regular old viruses that the rest of us have no problem with will kill these individuals. Okay. So if we could fix that problem, right, in their bone marrow cells, the beauty of your immune system is that it's built from your bone marrow. That's the site where all your blood cells are made. So red blood cells, white blood cells, platelets, all the stuff you think of that might be in your blood and your immune system is made by your bone marrow. Okay? And so most people that have SCID have a mutation in one gene that cripples their immune system. If we could fix this gene, then that bone marrow could then make cells for this person, and if that gene persisted in all of those cells, right, they'll essentially make a functional immune system the rest of their life, right, because you make blood cells every day, okay? So we replace our blood cells every day, make more, and so what we do is you can take bone marrow out of a person, right? You've heard of a bone marrow transplant, or you donate bone marrow, right? They usually get it out of your femur or your hip. That's where most of it is. So they drill a needle in there, which doesn't feel good. And they pull out some bone marrow, and then they can replace the defective gene with a functional copy of whatever they're missing. Okay? And then they can grow up those cells, make sure they're expressing the right protein, and then they jam them back in. As it turns out, if you just give somebody bone marrow into a vein, it will find its way into the bones and repopulate. Okay? And so now that person's bone marrow has a productive gene for whatever they were missing, and now they can start making all the white blood cells they didn't used to be able to make, and now they have a functional immune system, and they can come out of the bubble and be a normal person for the rest of their lives. Okay. So this is one instance where gene therapy works quite well. Again, sometimes side effects, right? And there have been instances where it doesn't work right and the patient dies, but this person is stuck in a bubble for their entire life. If they choose to come out and they didn't have any kind of therapy, they will die. Okay, so there was the story, there was an individual whose bone marrow transplant did not work and was done living in there and came out when he was 17 or so and died within 6 months. Right? So if they don't do something like this, they're either stuck in that bubble or they can't survive. So gene therapy will only work if it's really a one single DNA problem, one DNA mutation, one gene that you need to replace. If you have to replace a whole bunch of genes, which lots of diseases that we think about are a combination of genes, right? Like heart disease, there's not one gene that causes heart disease, right? But there is one gene that causes some metabolic disorders. People who have PKU, who can't, drink Coke because it has phenylalanine in it, that sort of thing, are potential gene therapy clients, okay? And so it's all about just giving them a new gene, replacing a defective one with a good one, okay? So here's a little picture. So we take out some immune system cells, which is essentially their bone marrow. You can isolate some of that from blood if you do a whole bunch of crazy stuff, but usually you just dig it out of the bone marrow, right? Again, use a virus to replace the normal gene, sticks it in there. We've made the virus non-virulent, so most of the time it's okay, and then we can give those cells back to the patient. And if it engrafts in the bone marrow and starts making new cells, then that person is cured. So because this still has lots of side effects and is very dangerous, we don't use gene therapy for things that are easily treated otherwise. So like PKU, for example, would be a candidate for gene therapy, except it's not life-threatening if you know you have it, and it's one of the things they test all newborns for, and you adhere to the right kind of diet, right? So the risk of doing gene therapy for something that's not a fatal disease is not worth it at this point, okay? Because it could kill you. But depending on the circumstance, if it's better than the alternative, then it's a candidate for treatment. So not something you just go into Kaiser and get for, some little silly thing. Okay. So that's what gene therapy is. It's considered genetic engineering because we're taking DNA and we're moving it from one individual to another. Okay. The process of cloning, of making an entirely new organism from a whole set of DNA, is also considered genetic engineering. Okay. We've been able to clone all kinds of animals, all kinds of model organisms. We know it works. It's not been done in humans. So there's essentially an embargo on cloning in human beings for ethical reasons. So a sheep was the very first organism to be cloned. Her name was Dolly. So they take the DNA. So like for human beings, it would be all 46 chromosomes from an adult cell, and then they take an egg, right, an unfertilized egg, take the chromosomes out of it, and stick the 46 chromosomes into the egg. So normally in fertilization, the sperm has 23 chromosomes and the egg has 23 chromosomes, and they come together to make the 46 for human beings. Okay. In this case, you just take out the 23 from the egg and put in all 46 from some individual, from some other cell. Right? If you take your 46 chromosomes and you put it into an egg, that's you. That individual would be identical genetically to you because it's all of your DNA. It's not like a normal offspring that half of the DNA comes from you and half comes from your mating partner, right? So it would be all from you. So that's what they did, and then they put that, so it's like in vitro fertilization, except there's no sperm. They use all the chromosomes from one other cell, and then they put it back into the sheep to go through gestation, and then Dolly, the sheep, was born. And she was genetically identical to her cloned mother. Okay, so they used 2 different species of sheep, okay, just so you could see phenotypically easily the difference between them. And so the real mom is the one that you get all the DNA from, right? So they take an egg cell from the surrogate mom, take out all the DNA, okay, and then put in DNA all 46, I don't even know how many chromosomes a sheep has, all the chromosomes from a sheep, right, into that egg, right? Now that acts like a fertilized egg. Okay, when all 46 chromosomes get in there, it turns on the machinery to make a new organism. It goes through embryogenesis, and a baby sheep was born. Okay, looks just like the donor mom, and when they look at the DNA, genetically identical to that donor mom. Okay, so that's a clone, right? Exactly the same DNA from the parent. Yeah, so his question is, well, what about a mutation that might happen? Could a mutation happen and so then they're not genetically identical? Yeah, definitely, right? Anytime you go from, so you go from that one fused cell, and then you go through however many rounds of cell division and mitosis, which we'll talk about in a couple weeks. Yeah, there's all this potential for mistakes to happen. And so, right, any small mistakes in DNA that happens during the process of going one cell to 80,000 kajillion trillion, I don't really know how many, but a lot, right, would be the potential for a mutation. And so those would be the places where they differ. So yeah, if you took and sequenced the entire, all 46 chromosomes, or however many there are, and all 46 chromosomes, and you're looking at the 46 billion different base pairs, yeah, there's likely to be a couple very small differences, just from random mutations that happen. But in general, you know, as exact as you can be. Okay. Let's watch a video. Do we think it's educational or not?
An exotic breed of cow has got everyone talking. It's a rare Asian calf known as a bantang, born recently at this Iowa research farm. The calf, just a couple of weeks old, is in fact making history. He appears to be the first healthy animal ever cloned from an endangered species. The story of this unique animal begins in California at the San Diego Wild Animal Park's Bantang breeding program. In 1980, an adult male died without producing any offspring. So, researchers at the zoo froze some of the Bantang's skin cells, hoping one day to put them to good use. They went into the frozen zoo, sort of a modern-day Noah's Ark. It's a deep-freeze menagerie of some of the rarest animals on Earth, 3,200 in all. Their genetic information is preserved in 4 tanks, kept at a minus 320 degrees Fahrenheit.
Cold.
Last year, the Bantang skin cells were removed from the frozen zoo and sent to the Advanced Cell Technology Lab in Worcester, Massachusetts. The Bantang's DNA was transferred into the eggs of ordinary beef cows. The fertilized eggs were then implanted into several surrogate mothers. While scientists believe this type of cross-species cloning holds great promise, it's not as simple as it seems. The process is challenging and unpredictable. 2 years ago, Advanced Cell cloned a rare Asian ox known as a gaur. It was hailed as the first birth of a cloned endangered animal. The gaur died less than 2 days after birth. Despite that setback, the scientists insisted the birth was proof cloning could work.
I think that the fact that we came this far is an accomplishment in and of itself. I think at this point it's just simply a numbers game. We know that in cloning, and it's just a matter of trying again.
Now, their determination and patience could pay off. The long-term survival of the baby Bantang would bode well for his and other endangered species, whose numbers could hypothetically be bolstered by this brave new experiment.
Aw, look at how cute he is. Okay, so that's really the idea of where cloning has gone. And so they're doing this with lots of different organisms. Of course, if we think about it, the species have to be relatively similar in order for, right, that egg that has the new DNA in it to go through gestation appropriately. Yeah. So the cloned animals, yes, should be able to reproduce normally. Okay, whether they actually do or not is still, there's not enough of them out there, but theoretically, yes. So it's not like a hybrid that we talked about where they have the wrong number of chromosomes and they can't get through meiosis to make eggs and sperm, right? They have their natural number of chromosomes, right, which should be even. They should be able to go through to make eggs and sperm. They should be able to mate normally. Okay. The one thing they do notice about cloned animals is that they age much more quickly than a standard offspring. And they've associated that with the age of the DNA that they use to make the clone. Okay, so as we age, our DNA does change and we have accumulated more and more mutations. But one of the biggest things that happens is that our chromosomes get a little bit shorter over time, okay, during the process of replication. And so they think that that has caused accelerated aging in cloned animals. And so what they like to do is the younger the animal they take the cell from to make the new clone, the better off that new cloned organism is. Okay? And so they have been doing cloning on endangered species, all different kinds, as long as, especially if there's some sort of agriculturally relevant stock to do the surrogate, like cattle, right? So any organisms that are related to cattle that have about the same gestation time, we can use those as surrogate moms, right? But you can't use, a cow as a surrogate mom for a sheep, right? So if we're trying to save the bighorn sheep, right, that may be endangered, we'd want to use a sheep species for that, right? The hardiest sheep species there is, it has about the same gestation time, right? You can't, if one organism's gestation is 9 months and another is 12 months, that's not going to work, okay? So you have to be pretty careful about that. Okay, and so sorry, this is just what I was talking about. So the cloned animals seem to age faster, and they're trying to figure out exactly why, but they think it's based on the age of the cells that they use. Okay, so if we think about Jurassic Park, right, and they took DNA from dinosaurs and they used frog eggs, and then they grew them in a fake uterus, right, or a fake egg, which you can't actually do. There's no organism has been gestated outside of another living organism to this point, okay? But the idea of that is possible if they get completely intact DNA from an extinct species, right? It can't just be some DNA and then they'll mix it up with frog DNA and then it fills in the empty spots, and then you get this amazing dinosaur that looks exactly like a dinosaur, not a frog, right? If it's dinosaur DNA and frog DNA, it's going to be a frog dinosaur, right? It would be a hybrid species. It would not look like the original. It probably wouldn't even go through gestation anyways, okay? And you'd have to find another organism that exists that could actually gestate or produce the egg or whatever for those organisms to survive. But it is possible. Right. And so when we talked about fossils, could you just find some old fossil and clone from that? No, because a real fossil is just rock, right? It's just minerals. You'd have to find like a frozen specimen or a mummified specimen in order to get intact DNA, right? So if sometime, in Antarctica, they're digging through the core of the ice and they find a T-Rex carefully preserved in ice, right, then it would be possible to clone that organism if we could find something large enough to gestate a T-Rex. Right? But a smaller dinosaur, right, you could use like the Gila monster or one of those monitor lizards or something like that and potentially do a cloning of an extinct species. Okay? So, that's enough of that. It's top hat time. Our favorite time of day. You're like, yes? Come on, people, work with me. You love this, yes. You guys are out cold. I've killed them. They are dead. All right, what is gene therapy? Gene therapy. Remember, all these points can go to any exam that you choose. Will be added to your total score at the end of the semester. Each question is a half a point for sending it in and a half a point for getting it right. So if you come every day and you do all the questions, you'll get at least 25 points of extra credit. And if you get any right, you can get up to 50 points of extra credit. People! But you have to be registered. So if you didn't register yet, you can still go register, pay your 20 bucks. And they will pull any questions you've already texted in or answered as a guest into your account if you request it nicely.
Do you call the number or?
I think you can just go online and contact their IT people, and then, yeah, they might say they'll contact you or you call them. But, yeah, they definitely, if you've been sending answers as a guest just because it's fun and you want your points, sign up. How are my texters? Are they going? Did they go? Are we good? Yes? People! What the hell? I have to make you guys all get up and do jumping jacks. All right, let's look at a report, shall we? Yay! So cloning is just called cloning. So that's easy. And that's all the DNA, right? Gene therapy is just one gene that you transfer into another organism. And we generally don't try to give people diseases, right? We might do this to a mouse in a study, but we wouldn't do this to people, okay? Oh, that's good. Good job, people. You guys are the best. No more answers for you. All right, go away. Go away. All right. All right, questions? Everybody's tired today? It's Tuesday. Okay. I'll do my best. Okay, so let's talk about therapeutic cloning, also known as stem cell treatments or regenerative medicine. Okay. So this is where we clone specific tissues or cells. Okay, and then we use them as a therapeutic, which just means as a treatment for some disease. Okay? So we're going to talk about stem cells. Stem cells are cells that can become other types of cells, right? In a human being, in an adult, right? Most of our cells are adult tissues. And we're not doing a lot of cell division. We're not making a lot of new cells, except in our bone marrow, right? Remember I said we're making blood cells every day, red blood cells, white blood cells. So bone marrow cells are considered stem cells. They're adult stem cells, right, because you're no longer an embryo. So we don't call them embryonic stem cells because they're in an adult, right? But they're definitely a tissue where they start as a bone marrow progenitor or a blood cell progenitor, something that comes first, and then they sort of grow up in the bone marrow to differentiate or become red blood cells. So the same cell in your bone marrow can become a red blood cell, can become the 20 different white blood cells, can become a platelet, right, from one cell. That one cell that can become all those other blood cell types is called a stem cell. Okay? That's an adult stem cell because it's in adults, and we are making those new cells every day. Okay? What normally we think about stem cells are the embryonic stem cells, cells that can become every other cell type in our body. They have to. We all started as one fertilized egg, one cell, right? Egg, sperm, woo, gets together, 23 plus 23 equals 46. That one single cell became you, your 20 gazillion billion different individual cells. Every single cell type in your body exists from one original cell. Okay? So that is the epitome of a stem cell. It can become everything, and it did become everything, because you're here, right? Okay. So that's an embryonic stem cell, right? The original fertilized egg can grow into an entire human being. Okay? They can make every single cell type. Okay. Yes, sir? So his question is, so hypothetically, if we took some stem cells and put it into somebody's leg, could they regrow their leg? It's within the realm of possibility if those cells get all the right signals that normally happen in development to make that happen. Okay, and so we'll keep talking about that and how we can regenerate tissues and what ones we can do right now and what tissues we're trying to work towards, right? Anything complex that has lots of different types of tissues, like a leg, right? If you think about a leg, it's got to have all the veins, it's got to have muscles, it's got to have all the nerves, it's got to grow a bone, and it's got to be in all exactly the right shape. It's unlikely that we'll be able to just stick stem cells in, an amputee's and have it grow a whole new leg, but it will be possible to grow the muscles, the vessels, the nerves, and all the stuff you might need, and then potentially put them around some sort of structure, right? Instead of growing the bone, you would use some titanium setup and build it around that mesh of structure, right? And so we'll talk about all the things we can do right now and then all the things that we're going to try to do. But first, let's talk a little bit about the process that will help you understand a little bit better of why some things seem impossible and why some things are definitely possible and are happening now, okay? And so what normally happens is the embryonic stem cells we get, we don't take the fertilized egg, okay, because that's one cell. We let that grow up to a stage of about a ball of 8 cells or so, okay? And because it's much more stable. So these embryonic stem cells that are used for treatment and research come from in vitro fertilization attempts, okay? And so let's first talk about in vitro fertilization and then talk about how that gets to a research lab or to the clinic. So in vitro fertilization is just something that couples can do if they can't have a baby the good old-fashioned way, okay? There's some problem with ovulation, or there's a problem with the sperm swimming, or there's some other issue that's going on, and couples are infertile, they cannot have a baby, right? We can take the eggs from a female and the sperm from a male and do the fertilization event in a Petri dish. So in vitro just means like in a test tube, outside of the body. Okay? So we can, instead of it happening up in the fallopian tubes, right, we can do this in a Petri dish. Okay? And so the process would be in the females, they take a whole shitload of fertility drugs so that they superovulate a whole bunch of eggs, and then those eggs are surgically removed, right, and put into the Petri dish. The sperm is much more easily acquired, just a magazine maybe and a closed door. Okay, that goes in here. It's in nutrient media, right? The sperm do their job, get in there, fertilize those eggs, and then these are let to replicate until they get to a decent size. It looks like they're going to continue development, and then they're reimplanted into the female. Okay? Usually, in vitro fertilization, now they're getting really good at making it work. No more than 3 embryos are put in at any given time, right? Because the human beings are supposed to have 1 baby, right? Sometimes twins. The occurrence of a natural triplet that's not been messed with any drugs or something is extraordinarily low, but it is possible for a human being to have 3 in just by good luck or bad luck, however you might want to look at it, okay? But extraordinarily rare. Any more than that is not acceptable for people, right? We're not supposed to have litters, right? Animals that have a litter have uterine horns, which are very long, and they're set up to have 6 on each side, right? We're set up to have 1 baby in there, 2 at the most. 3 becomes usually dangerous for the mother and the offspring, okay? So they try to not put more than 3. If the person's relatively young and healthy, they'll only do 2 in the first round. So lots of in vitro fertilization results in twins, okay? So if they put in 2 and they both stick, you get twins. If they put in 1 and it splits, then you have identical twins, okay? And in vitro fertilization, the event of identical twins is much higher than in the good old-fashioned way, right? So the identical twin population has risen due to this, right? So if they put in 3 and they all stick and 1 splits, then you have 4. And that could be, or more than 1 splits. And then that becomes dangerous to the mom and to the babies. So we're going to watch a little video clip. Do you think it's educational? Oh, you guys are brilliant. Okay. So, let me say one more thing. So if the problem is something with the sperm, right? So in a lot of cases, the egg was fine, but there's a problem with the sperm. Instead of just dumping the tube all over the eggs in here, they can actually inject a single sperm into an egg. So if the problem is the sperm can't get in the egg, like that's its malfunction, then they can actually poke it in there and help it out. Okay, so the little video, I think, is going to show us that single sperm injection, which, as it turns out, is a lot more expensive than when they just dump it all over the eggs in there. But it works quite well.
In preparation for in vitro fertilization, a human oocyte is held in place. In this technique, intracytoplasmic sperm injection, a single sperm is drawn into an injection pipette. Gentle pressure is applied to break through the tough outer cell layer and insert the pipette into the cytoplasm. Once inside, the single sperm is released and the pipette is carefully withdrawn.
Pretty awesome. So then if that goes through, right, the proper development, it will become that ball of cells, and then we can implant that into the uterus, right? If we know anything about reproduction, right, the egg and the sperm normally meet in the fallopian tubes. It takes about 7 days for it to travel down to the uterus to implant, okay? And so in that period of time, it's replicating. So the implantation event is usually between, 8 cells and 16 cells when it actually implants. So it's about a week after fertilization. Okay, and so they try to mimic that in the same way doing in vitro. Any fertilized embryos the ball of cells that look like they're functioning healthy, that are not being used, are frozen. So they can be frozen same with in that minus 300 and whatever Fahrenheit liquid nitrogen tank, just like the zoo, they said, right? So the same kind of cold, those embryos can be frozen and saved for later. So let's say they do the implantation, couple has a baby or two, 3 years later, they're crazy enough to do it again. They go back into the freezer, they pull out 3 more, they do that. At some point, after they've had enough children, they're done. Okay? At that point, those embryos that are left over are in the freezer. And at that point, the parents are given the choice, what do you want to do with these embryos? Do you want to keep them frozen forever, which costs you $1,000 a year or something? Do you want to donate them to another couple that's infertile, that their in vitro didn't work for whatever reason, the egg and sperm were so messed up they couldn't make it happen, right? Or do you want to donate those to research? Or do you want to throw them in the trash? Okay, so those are the choices. So it's up to the parents whether these fertilized embryos go to research or not. Okay, so they have to be informed consent and all sign a million papers and so forth. Okay, so that's the choices when somebody has leftover IVF. So that's where the embryos come from to do any kind of embryonic stem cell work. Yeah?
Now, when it comes to abortion, when is this considered abortion? At what stage?
So her question is about abortion, and what is this considered, would it be considered abortion in like a real person?
Yes.
So at the point of fertilization and implantation, right, you have no idea you're pregnant, unless you're really, in tune. Your pregnancy test does not become positive until a week after implantation. Okay, so no. So that at the stage, once they're good enough or more developed enough to have a good implantation, you can no longer get embryonic stem cells from them. Okay, so they've gone through that stage where you could get embryonic stem cells. So if somebody peed on a stick, has a positive pregnancy test, and then runs and says, I want to donate this to research, it doesn't work. It's already too late. Okay, so you can only get those cells before anybody knows they're pregnant. Okay, and they're never taken from a real person or from any aborted fetal tissue. They can use fetal tissue to do therapeutics, right? Like or miscarried, however you want to say it. If you donate that tissue, right, just like being an organ donor, if you had, a miscarriage and you were at the hospital, you could donate those tissues to be for organ donor. So usually they're used for cornea transplants, right, eyes, occasionally for liver, anything that you don't need an entire organ for, right, because the fetus is way too small to like do a liver transplant or something like that, but you can get tissues from them to do some therapeutics. But again, that's by parental consent if they want to donate that or to research to study those types of transplantations. So yeah, so to get embryonic stem cells, it cannot come from any person. It has to come from an in vitro fertilization. Okay, and right now, you can't do in vitro fertilization just to get embryonic stem cells to do research. They have to come from a donated IVF lab. So just because you're in a lab, you can't say, okay, I want to donate my eggs, and then my lab partner is going to donate the sperm, and we're going to make some embryos, and then we're just going to study them. You can't do that either. Okay, so they have to come from donated individuals. And for the most part, that's relatively rare now, because what we've been able to do is take those embryonic stem cells from those few embryos that they get and create what you call a cell line, grow them in culture essentially forever, right? Because they're stem cells. Their thing is to make more of themselves. And if you grow them in culture under the right conditions where you're not giving them any developmental signals, they will stay as embryonic stem cells. They won't differentiate into any of the other tissues, right? So you can grow up 1 billion of them take a couple 100 million, do your experiment, and keep growing those cells so that you don't go back to a fertility clinic to get more embryos all the time. Very, very few labs actually get live embryos now to extract the cells from. Right? There's a couple labs in the whole country that does that, and they provide the cells to every other researcher in cell lines. Okay? So it's very rare to actually use a real embryo anymore. So most of them are these cell lines that they're using. And then those you can freeze down just like any other types of cells. So we use cancer cells in labs to study things as well. Those can be frozen, so you can keep them forever, bring them back out, grow them up, and that sort of thing. So you can perform all the studies without ever destroying any embryos. Okay, and so then this would be the overall idea of stem cell therapy, right? So you grow, you do the in vitro fertilization that's donated. You get to the point which they call the blastocyst, and that's the thing that implants into the wall of the uterus, right? And then that little mass that's inside, that's the stem cells. That's what becomes the baby, okay? The ring, the cells around the ring, that becomes the placenta inside the uterus, okay? And so what happens is those, as soon as implantation happens, those cells start developing into the fetus, right? So right after implantation, they start changing. They start making the different layers, right, to make muscle and bone versus, you know, all your inner organs, the ectoderm, endoderm, mesoderm, all that baloney during development, so that by the time the person knows they're pregnant, that's already been developed beyond the stage where you can get those embryonic stem cells out. Right? So they have to be very, very early, before, essentially at implantation or before, in order to get those. Okay? And so if you take out that mass of cells, right, we can grow them up, right, make a cell line out of them and keep them growing forever. Take some of those cells, we call those pluripotent stem cells. That means they can become almost everything in the body. The only thing those cells can't become is the placenta, because those cells were already differentiated into that outer ring. So pluripotent just means become every different cell type except the placenta. Totipotent, or totally potent, means it can become every single thing, including the placenta, and that would have to occur at the fertilized egg stage, or before it makes that little ring out there. Okay? So embryonic stem cells that are used for therapeutics are pluripotent. That just means almost everything can become almost everything. Everything we need, like especially boys, right? You're not going to need a placenta, I hope. Okay? Right? So every kind of cell type that we need. We know, scientists know, I don't know, all of them, how to differentiate lots of different cell types from those embryonic cells. The exact nutrients, hormones, proteins to put into a flask to grow nerve cells, to grow heart cells, to grow more skin cells. Okay? We can grow almost every single cell type out there now, or very close to it. Okay? We can do this in mice. Right, the same idea, in vitro fertilization for mouse, grow up some nerve cells, right? Take a mouse that has a spinal cord injury, inject nerve cells into the spinal cord, they engraft, they heal, the mouse walks again. We have cured paralysis in model organisms using stem cell therapy, right? That's one step away from doing that in human beings. That's power, right? We can make heart cells in mice, and we can give them to a mouse that had an induced heart attack, where there's a lot. Heart attack is just dead heart tissue, right? If too much of your heart is dead, guess what happens? Yeah, you're dead. Okay. So if you survive a heart attack, it's because not enough of your heart tissue died to actually kill you. It can still function, right? It's never going to be the same if you didn't have a heart attack, because there's a patch of cells that are dead. Right? We can take in a mouse, we can take those heart cells we made in culture, jam them back into the heart tissue. The heart's actually in this little membranous sac. You just fire them in there. They engraft. They heal over that scar tissue. Mouse as good as normal before the tissue. One step away from doing it in people. Okay? And about $4 billion. Yeah. So that's a great question. So is this why sometimes they take the cord blood from the baby when you give birth? Yes. So cord blood only has the bone marrow stem cells in it, okay? Because it's actually very rich in that. So the baby's bone marrow and the bone marrow actually is in that cord as well. And so they save that, and that's like saving extra bone marrow for that child, right? And so that if that child needs a bone marrow transplant later or needs gene therapy done, something like that, then yeah, they can take that out of the freezer, manipulate it, and put it back into that child. The question is, can they use that for the parent? Let's say something happens to one of the parents. It's not as helpful because your immune system will see it as foreign because it's not yours, right? And so that's the problem with any kind of transplantation. Like if you know somebody who's had a kidney transplant, they have to take immunosuppressive drugs the rest of their life because their body will reject it as foreign, right? Your immune system job, anything that doesn't belong needs to get kicked out, including cells, and that's bacteria, viruses, you know, to keep us safe. So we have to kind of ramp down the immune system in order to keep any kind of transplant that didn't come from you, right? So even if it's your kid, they should be about half as good as you, right? But still not, sometimes not a great match, especially if your partner is totally different than you, and right? You're better for transplants as a sibling. You're actually more genetically similar to your siblings than to your offspring, right? Because you shared the same mom and dad. You didn't get the exact same half of dad and half of mom, or you'd be unless you're identical twins, right? But it's usually much closer than a parent to a child. Okay, so let's watch this, another educational video. What the hell's happened to me today? Oh no, I'm off my net.
Dr. John Gearhart was at the forefront of that achievement.
These cells have two properties. One is that if you keep them in the dish under certain culture conditions, they will continue to form more cells like themselves. So you can grow a room full of these embryonic stem cells, and they are undifferentiated cells. They all look alike. If you take some of these cells, though, and you put them out in different kind of growth conditions, these cells are capable of forming all the cell types that are present in the human body.
What we are looking at here are heart muscle cells that are beating as a tissue.
Once, these were stem cells with uncharted destinies. Dr. Gearhart has directed their development into heart cells, now able to beat in perfect synchrony.
Yeah, that's what I'm talking about, people. Okay, so why can we do this? Why can we do this with a mouse, right? A lab mouse, and put the cells back in, and it cures their heart disease, and they're fine? Like, why don't they reject the cells? Because mouse lines are so inbred, the ones we use in labs, they're essentially clones of each other. Okay? They've been inbred for so long that their DNA is almost identical, and so between two different lab mice of that same strain, they don't reject each other's tissues because they don't see it as foreign. Okay? So the biggest problem with embryonic stem cell therapy right now is that it's not you, right? Even if it's half you, it's not you. That tissue, we grow up and make heart tissue for you, even if it was from your embryo, right? It's not going to be, your immune system is going to still see it as foreign. Okay? So the proof of principle that this can work is established well in these model systems where all the organisms are essentially clones of each other. right? So we can avoid that whole immune rejection. So we know it can work if we can just get to the point of no immune rejection, right? Somehow do that without using, somehow make the same kind of embryonic stem cells, but not from an embryo that's not you. Okay, and so the biggest problem with stem cell therapy, even though it's got this enormous potential to do all kinds of amazing stuff for almost any disease you can think of out there, is that the federal government is scared of it. Okay? It smacks of very, very science-y. And science-y stuff and big words scare politicians. Okay? And they also have problems with religious groups who are under the assumption that scientists are running around having women get abortions to steal babies to do stem cell work, which, as we all now know as educated Bio 100 students, you cannot get embryonic stem cells from any aborted tissue. Okay? You can only get it from the fertilized egg that's grown before it can implant into the uterus. Okay? And they all come from donated embryos that are left over from people who no longer need them. Okay? But the whole when does life begin and when is it murder and so forth is a huge issue, right? A huge ethical issue, and politicians are scared to generally make a stand one way or the other. Okay? So the Republicans make a stand and say, no embryonic stem cell research is allowed. We're not going to fund it by the federal government. Okay? The Democrats, generally a little more liberal, right? That's our thing. And they say they're going to allow some of it, but then usually Congress says no. So up to this point, there's been very little federal funding for any kind of stem cell research. Most medical research that happens in this country is funded by the government, the National Institutes of Health. That's who we write grants and beg them for money and try to convince them that what we're doing is good. Okay? So what happened is the states took it into their own hands, and several different states came up with their own funding sources to do stem cell research. Okay? So, no, I'm lying. So, in California, the California Institute for Regenerative Medicine was passed on the ballot in 2006, and we all voted, well, you guys were probably too young, for $3 billion in bond funds to go towards stem cell research in California. And so California, Massachusetts, I think Connecticut, and a couple other states have the same, Michigan maybe, the same types of funding so that we can progress in stem cell research. Okay, so this funding source gives out a lot of money. As it turns out, we here at CSUN have a bunch of money from them. And so through a grant that I wrote with my friends at UCLA, we have $3.4 million to do stem cell research at CSUN. And so what we do with that money is we send our students over to UCLA to use all their good stuff and all their expensive stuff, right? So we're totally like mooching off of them because they have it all set up. They have an entire building built with this CERN money, right, with all the high-tech, most expensive equipment that you could get, and so we just mooch, right? We send our students. I send them with a whole bunch of money, right? They get paid a little stipend. They get their tuition paid, and they go over there and they work their butts off, okay? And so what a lot of the students are doing, one of them was working on this mouse project with the heart cells, right? There's a couple students that work on the mouse and the paralysis. There's Parkinson's disease. There's all different metabolic disorder stuff that they're working on. And one of the biggest things that they're doing is not only are they looking at the embryonic stem cells, right, which can become anything. One of the, so here's just a little review of the different types of stem cells. So if you're a single-celled fertilized egg, that's the only thing that's totipotent, right? Can become everything, including placenta, right? if you're this 5 to 7 day old embryo with the ring around it and the inner cell mass, the ring is already going to be placenta. The inner cell mass is now pluripotent, become everything except placenta. Right? If you get cord blood or adult stem cells, these are considered multipotent, right? Because those can only become more blood cells. Right? They're from your bone marrow or the cord. Right? They can become red blood cells, white blood cells, platelets, but they can't become nerve cells. They can't normally become heart cells or any of the other things in their normal differentiation. Oh my God. So everybody's good with totipotent versus pluripotent versus multipotent, right? So think of as multi as most, pluri is almost everything, and toti is toti, total. Okay. We also can find some stem cells in lots of different tissues, right? They're really, really difficult to isolate, so it's not like something we just stick a needle in and suck a bunch out like we can with bone marrow, right? But there are scientists trying to isolate, well, the bone marrow is easy, like heart cells that are already the stem cell in there, right, to use. Because then that way you could take out your own cell, grow them up in culture, and then give it back to you, right? Then you wouldn't have any rejection problems. But that's really, really difficult. They're really, really hard to find. The best thing that's out there now, the best hope for therapies is something called an induced pluripotent stem cell. This is what lots of our students from CSUN are working on right now over at UCLA. This is where you can take a skin sample from somebody. So I just come over and I just punch biopsy, right? Just take a little chunk of your skin out. And you're like, what the hell? And then I stick it in culture, right? And I essentially do gene therapy on it. I give it a bunch of DNA protein factors that have it reprogram itself and it, oh my God, I think I'm a stem cell. Okay? And those cells actually change what proteins they're making, right? Their gene expression, they turn off their skin cell program and they turn back on, I'm an embryonic stem cell, I can now become anything. Then we can take those guys and drive them to become heart cells or nerve cells or skin cells or pancreas cells to treat diabetics. Okay? It's your own cells. You won't reject those because they're yours. Your immune system will see it as you, right? So if we grew up heart tissue for you because you had a heart attack, we can just jam it right in there. Well, it's not that easy, but right? And we will not have an immune rejection. It's your cells. Right? This is where the therapies will come from. Okay? So we can do this in mice and all the model organisms and grow them up, and yes, it does repair nerves damage, spinal cord damage. Yes, you can have them engraft into heart tissue and help with heart disease. Yes, you can cure a diabetic mouse by doing this process. So again, this is an even closer step to doing this in human beings. There are some clinical trials now doing this for paralysis. Okay? Clinical trials are really, really expensive because you want to make sure everything's perfectly safe. They do all the tests in the flask first, and then in an animal model, and then in people, right? And the people obviously have to be volunteers, right? So they're volunteers, they know exactly the risks and the possible benefits, and they're willing to take those risks to do those types of clinical trials, okay? The biggest problem with the iPS cells is the gene therapy part, is getting that DNA into those cells safely. Okay? And so what we found is, right, we introduce these genes, treat with small molecules, right, grow and differentiate, we get all of this stuff, we put them in animal models, and maybe 1 in 100 of those mice that we treated get cancer. Because those cells started growing out of control. right? Cancer is just cells growing out of control. 1 in 100 mice, not a problem. 1 in 100 people, not a good number, okay? And so that's the problem as far as getting from the lab to the clinic, is solving that problem of how can we avoid using viruses here, right? Or this process of, are all of these now really nerve cells? There's nothing that's still a stem cell that could become something else like a tumor, right? That's where we're stuck right now in therapy to get to human treatments, okay? And so just like the human embryonic stem cells, right, we can get these induced pluripotent stem cells from any adult tissue, turn it backwards, make them think they're embryonic stem cells, and then grow them into anything else. It's the safety issues that are being dealt with now. All of those things are really expensive. All of those things take lots of money, and the federal government is still not funding it. Yeah. His question is, what causes cells to grow out of control? It's usually some kind of mutation that happens, usually. We're going to start talking about cancer in the next couple weeks, and we'll talk about how mutations can happen, usually in a specific type of gene or more than one that is supposed to be controlling the cell division, and it gets out of control. That's why it can be in all different types of the body. That's why cancer can happen to any cell. It's just one of your own cells that got out of control, right? And then it keeps going and going, and a tumor is just a mass of cells. And we'll talk about why a mass of cells can be so dangerous. And so here's, yay, all this stuff that's going to cure everything, and we need more money. See, all this good stuff, yay.
Right, okay, people, you have to tell me these things, all right? I've been frozen for 30 years, okay? Throw me a frickin' bone here.
Yeah, like that. Okay. All right, let's do one more top hat. Get you guys some more points. I'm all about getting you guys some points. Okay, so we've been over this a bunch of times. This is true or false. All the different types of stem cells are totipotent, meaning they can become any other type of cell in the body. Holy crap, we went from 0 to 100%. Did you see that? You guys are fast. Ah! All different types, all the ones I talked about. The bone marrow ones, the embryonic ones, the IPS ones. Remember, IPS stands for induced pluripotent stem cell. Hint, hint, hint. Totipotent meaning every single thing, including the placenta. Any other questions while we're firing away our texts? Yes? No? Yes? Go! Are we ready? Are texters ready? All right. They're just ignoring me. Fine, be that way. False! Oh my God, people! Ah! They're not all totally potent. What does pluripotent mean? Can become almost everything. What does multipotent mean? Become some things. Right? You get it?